Okay, let's dive right into this explainer. Right now, we are facing an absolute mountain of biological data. I'm talking genomic sequences, spatial transcriptomics, unbelievably complex protein structures. It's just a continuous torrential flow of information. And to actually survive it and make sense of it all, we need incredibly intelligent ways to sort, analyze, and truly understand what we're looking at.
So, imagine this massive ocean of data. Some of it is perfectly labeled, some is completely unlabeled, and a vast majority is just kind of sitting somewhere in between. Unlocking its secrets really comes down to choosing the exact right machine learning paradigm. Basically, there are three fundamental ways we can teach machines to learn from all this.
Let's start with the first one, supervised learning. Think of this kind of like a student who already has the answer key. The algorithm is handed the absolute ground truth right up front. We feed it fully labeled training data, and from that, it learns to perfectly map inputs to specific outputs, drawing these dynamic decision boundaries so it can classify data it has literally never seen before.
To actually pull this off, we bring in the heavy hitters, things like support vector machines, random forests, and neural networks. These are the powerful engines we use when we know exactly what we're looking for, allowing us to hit known classifications with incredibly high accuracy.
And this is the exact engine behind some truly massive breakthroughs. For example, AlphaFold can predict complex 3D protein structures, specifically because it trained on huge databases of known proteins. Or think about diagnostic models. They learn from thousands of known cancerous tissues to instantly flag new patient samples. It's a real game changer.
All right, now let's completely strip away those labels. Enter unsupervised learning. This is basically the student figuring out the math entirely on their own, without a teacher. Handed raw, completely unlabeled data in the wild, the algorithm's job is to organically discover all the hidden patterns and secret groupings buried inside.
Here, our go-to exploratory tools are algorithms like k-means and hierarchical clustering, which essentially group data organically by shared traits. We also use principal component analysis, or PCA, which reduces the noise so we can actually visualize these wildly complex dimensional relationships without any preconceived human classes getting in the way.
So where does this really shine? Discovering the completely unknown. When you feed it unlabeled single-cell RNA sequencing data, unsupervised learning organically clusters distinct cell types in a tumor microenvironment. It reveals entirely new patient stratifications that we literally could not have found manually.
But, there's always a catch, right? The crucial gap between the two is that supervised learning requires incredibly expensive, grueling, expert annotation to get those labels. On the flip side, unsupervised learning is entirely free of labels, but it can lack the targeted, predictive direction we really need in high-stakes clinical settings.
Well, enter semi-supervised learning. It's essentially the perfect hybrid. The algorithm learns a few core concepts from a tiny, super affordable batch of labeled data, and then it goes out and practices on a massive ocean of unlabeled data to build a remarkably strong predictive model.
Here's exactly how that flow works. First, you train it on that small labeled batch. Next, you let the model predict labels for the unlabeled data, creating what we call pseudo-labels. Finally, using a technique called label propagation, it takes its absolute highest confidence predictions to relentlessly refine itself. This is exactly how we tackle massive non-coding RNA datasets or protein annotation, where full manual labeling is just downright impossible.
This perfectly synthesizes the whole journey. Your input data entirely dictates your approach. If you need high accuracy for known outcomes, you absolutely need supervised learning. Have zero labels but need to discover hidden patterns? Unsupervised is your best bet. And to maximize your predictive power when labels are super scarce, well, semi-supervised is really your only path forward.
So, if you take absolutely nothing else away from this explainer, let it be these high-contrast definitions. Supervised is for prediction, unsupervised is for discovery, and semi-supervised is your bridge when labels are limited but your data is abundant.
Now, what's incredibly fascinating is how we apply all this to high-stakes biology. For instance, using graph neural networks, we can map spatial transcriptomics, clustering and analyzing human cells exactly where they sit in the tissue. We're identifying unique cell types and segmenting subcellular patterns in this continuous, beautiful motion of discovery.
Because really, these aren't just abstract algorithms anymore. They are the new engines of genomic medicine. From tracking the real-time evolutionary dynamics of invasive melanoma to predicting life-saving 3D structures with AlphaFold 3,
These paradigms are actively turning static data into dynamic, actionable cures.
So the next time you face your own mountain of biological data, you have to ask yourself one simple question. Do you want to predict the known or discover the entirely unknown? The labels you choose to use or choose to ignore might just hold the key to the very next great medical breakthrough.